Jim Allison. I'm the chairman of the Department of Immunology and director of the Immunotherapy platform at the MD Anderson Cancer Center. As an immunologist, you're so close to medical issues, and especially cancer, that you have to be thinking about it. In my case, I thought about it a lot because my mother died of cancer when I was about 10 years old. But when I took my first faculty position, I decided I was really intrigued by the immune system and trying to learn how you could have this wonderful thing of cells and soluble molecules that go throughout your body looking for things that have gone awry and manage to protect you and not destroy you somehow. And so I was really curious how that worked. So I basically spent almost my whole career in immunology trying to understand what regulates T cells, what turns them on, what turns them off, just exactly how they work, and molecular mechanisms of it. By the late 1980s, it was clear that the antigen receptor, which is kind of like the ignition switch on a T cell, the structure of that I worked out in the early 80s actually. But by the late 80s, it became obvious that that wasn't enough. Just having that on, it wasn't just an on-off switch. There are other signals, and so one of the first things we did was to, in that area, was to identify what was called a co-stimulatory receptor, this molecule called CD28. It's kind of like the gas pedal. You have to get a signal through it to actually get T cells to start doing something. And then there was this other molecule that had already been cloned that was very highly related to CD28 called CTLA4. And the question was, what did that do? And it was very controversial. But a colleague of mine at the University of Chicago, Jeff Bluestone, and my lab both proposed that it was actually not another gas pedal, but it was the brakes. And there was this program that was hardwired, to turn on the brakes gradually and stop the process. Then I had the thought, well, I wonder if we could make the immune system better at attacking cancer by just temporarily disabling the brakes, basically keep the immune system from turning itself off for an extended period of time until it had time to eliminate tumors. And that was the basic idea. It was understanding the basic biology and then making the leap to say, well, we can manipulate this to our advantage to treat cancer. And it turned out it worked. When it really, I guess, had the impact on me was when I started meeting the cancer patients. My colleague Jed Walchuk, for example, called me. It was in the early days, and to meet him in the outpatient center at Memorial Sloan Kettering when I was there, and to meet this young woman that had just been told that she'd been free of cancer for a year after getting ipilimumab, the drug that I helped develop. And that was an incredible moment.